Welcome back to this next video in which we are discussing coronary artery disease. Okay, so we are now going to discuss then the consequences of having an atherosclerotic plaque in one of your coronary arteries. And for the purposes of having an example, let's just suppose that it is within the left anterior descending artery. Okay, so we're going to suppose then that we've got an atherosclerotic plaque having developed over decades in the left anterior descending artery, and this is of course going to reduce the diameter of the lumen of the blood vessel. And what we want to now understand is what are the consequences of that? What are the consequences of coronary artery disease? And the chronic complication of it is something known as stable angina, okay? Or to give it its full name, stable angina pectoris. So let me put this as a title and then I'll describe what it actually is. So stable angina, and then if we're going to give it its full name, we should add on here pectoris, and we will come to the other one, which is unstable angina, and we'll contrast that. Unstable angina is an acute coronary syndrome. It's the least severe of the acute coronary syndromes. It's the same pathogenesis as a heart attack, however it's where it's not quite as severe and you haven't actually had cells die yet, but we'll talk about that later on. So at the moment, let's talk about stable angina pectoris. Okay, so firstly, let me describe what actually is stable angina pectoris. So we'll start by defining it, and then we'll understand why having this atherosclerotic plaque in your left anterior descending coronary artery, for instance, is going to actually lead to stable angina pectoris. So, a bit of Latin then for you now. Angina, this comes from the word angere in Latin. And angere in Latin is apparently a verb, and it means to strangle. So here is the infinitive, I believe. I'm not an expert in languages, but I think this is the infinitive of the verb. So to strangle, the infinitive form of the verb. So angere is the verb to strangle. So angina comes from that, and we'll understand why we're describing this as a strangling in a moment. And the pectoris here comes from the word pectus in Latin, which is a noun and means the chest. Okay, right, so these are Latin words then. So angina pectoris, strangling on the chest, or strangling in the chest, and that nicely describes what actually happens in stable angina pectoris. Stable angina pectoris then is the condition where you get a crushing or strangling or horridious chest pain upon exercise. So you're fine when you're at rest, you're absolutely fine when you're sitting down on your sofa or whatever, watching television, you're absolutely fine, no pain then. But if you get up and try and jog or exert yourself in some other way, you're going to get this crushing or strangling chest pain. This is how people actually describe it. I've obviously never experienced an angina pectoris attack, but people describe it as a strangling, crushing chest pain, as though someone is trying to strangle their heart. Okay, hence why it's described as angina pectoris, strangling of the chest. Now, it also often leads to referred pain up to the left shoulder. That's the common referral site for the pain, and I'll explain why you get referred pain to the left shoulder later on. Okay, so you can also feel pain up in your left shoulder. These are the classic descriptions of someone suffering from an attack of angina. Now, the stable portion here in the name, that denotes the fact that this is only brought on by exercise. So when you are sitting in your chair resting, you are not suffering from this crushing chest pain. In contrast, in unstable angina pectoris, which is going to be an acute coronary syndrome, you're going to have the crushing, strangling chest pain when you are sat down and at rest. Okay, so that's the difference between stable angina pectoris and unstable angina pectoris. Stable angina pectoris is just crushing chest pain when you exert yourself. So when you try and exercise, you're going to get this crushing chest pain. And this is the chronic condition that coronary artery disease, having an atherosclerotic plaque in your one of your coronary arteries, is going to lead to. Whereas that atherosclerotic plaque rupturing, that's then going to lead to the acute coronary syndrome, which, if you're very lucky, will just be unstable angina. If you're not so lucky, it will be a heart attack, and we'll come on to those later on. Okay, so in stable angina pectoris, you just get this crushing chest pain when you exercise. So what I now want to explain then is exactly why having an atherosclerotic plaque in one of your coronary arteries, so the example that we've taken is the left anterior descending coronary artery, why that's actually going to lead to getting crushing chest pain that refers up to the left shoulder when you exercise. Okay, so remember that having this atherosclerotic plaque in your coronary artery is of course going to be narrowing it. Now, when you have a narrowing in a blood vessel, what is that going to mean? I'll just draw a picture here. So let's say we've got a cross-section of our blood vessel here, and this is a different cross-section to the cross-section we drew before. This is now a longitudinal cross-section rather than a transverse cross-section. Okay, so with the length of the blood vessel. And here is our atherosclerotic plaque, clearly here, reducing the lumen of the blood vessel considerably. And what's going to happen? Well, of course, you're now going to be trying to squeeze all the blood through this narrow lumen now. Okay, so that means that when the blood actually squeezes through there, of course, it's going to now pass much more fastly. I hope your intuition about fluid dynamics is good enough here to understand this. If we're trying to get a certain amount of blood through here, we're going to be pushing it through a smaller pipe now. It's going to have to be moving faster. So indeed, when the blood goes through here, it's going to be moving faster because of the narrowing in the blood vessel. So if you're imagining trying to squeeze blood through here, when it's actually going through that narrow portion, it's now going to be moving past the endothelial cells here faster. Now, what happens when blood moves past endothelial cells at a raised rate? Well, of course, the endothelial cells get activated to send a signal to the smooth muscle cells to tell them to relax down. Okay, this is a very classical pathway that medical students everywhere must learn. So, let me draw it out. So, if I draw one of the endothelial cells over, of this portion of the blood vessel where we've got the narrowing, so let's say here, and I'll draw it a little bit bigger than that, so this, let's say, is a strangely shaped endothelial cell over this portion of the blood vessel here. Okay, so it's sitting on its basement membrane, which I'll draw in green here. So, when it's now got the blood passing over it at this increased rate, what that is going to lead to, the fancy way of saying this, is it's going to lead to an increased shear stress. Now, shear stress means the force that is on the endothelial cells. So when you've got blood moving like this, it's going to create a force on the endothelial cells, a friction if you like. Remember, the blood's made up of loads of little molecules, the endothelial cell is made up of loads of little molecules. These molecules will be smashing into each other, and you're going to get a force being exerted on the endothelial cell. That is what is meant by the shear stress on an endothelial cell. Now, when the shear stress of on an endothelial cell is increased, what does that generally mean? Well, that generally means that the blood vessel is too narrow, because it generally means that the blood is having to move through the blood vessel too fast. So what is the solution to try and reduce the speed at which the blood is moving past the endothelial cells? Well, of course, it's to try and vasodilate, try and increase the diameter of the lumen. So there is a pathway that all blood vessels have, a signaling pathway that is installed in endothelial cells that allows them to tell smooth muscle cells of the artery wall to relax and increase the diameter of the blood vessel if the shear stress is too high. Okay, and they release nitric oxide, or the endothelium-derived relaxation factor, and this process is known as the endothelium-derived relaxation pathway. Okay, so this is the pathway that I'm just briefly going to remind you about. So when we've got this stenosed blood vessel here, and we're now using that fancy keyword that I stated at the start, so we've got a stenosed blood vessel, a narrowed blood vessel due to the presence of the atherosclerotic plaque, we're going to have increased shear stress on the endothelial cells, and when you have increased shear stress on endothelial cells, there is a very famous physiological pathway for the endothelial cells to go down in order to try and reduce the shear stress on them, in order to try and reduce the speed at which the blood is passing over their apical surfaces, and this involves them telling smooth muscle cells of the artery wall to relax. Okay, so what happens is endothelial cells that are feeling increased shear stress start releasing the signaling molecule nitric oxide, NO, which is actually a free radical. It's made up of a nitrogen atom double bound to an oxygen atom, but of course nitrogen likes to have free bonds usually, so one of the electrons of nitrogen is actually unbound, so it's on its own, it's not in a pair basically. So this is the molecule nitric oxide. It's not a particularly reactive free radical. So this is nitric oxide. So the endothelial cells release nitric oxide, and they release it in response to the increased shear stress. And we're not going to go through what is understood about the signaling pathways by which increased shear stress actually causes an increase in the nitric oxide synthesis of the endothelial cells, pardon me. It's not particularly well understood how the endothelial cells actually detect the increase in shear stress and then increase their nitric oxide production. So we'll just do this on the cellular level. When the endothelial cells sense the increased shear stress, which means the blood must be moving over their surface at this rapid speed, they're going to start releasing this molecule nitric oxide, and nitric oxide is a very small molecule. It can diffuse through the cell membranes. It'll diffuse back to the smooth muscle cells in the tunica media, deeper down in this blood vessel wall. Okay, so now let me show you what it's going to do in the smooth muscle cells. So this pathway is very important for us to understand so that we can appreciate later how the nitrovasodilators, which are used to treat asthma, are going to actually work. Okay, so here is our vascular smooth muscle cell here, and I want to just draw out the little pathway by which nitric oxide actually causes vascular smooth muscle cells to relax down. So nitric oxide will arrive in the vascular smooth muscle cell, and there it's going to activate an enzyme, which I'll draw here, like so, which is known as the soluble guanylate cyclase enzyme, and for short this is abbreviated down to SGC. So this stands for, the little s is for soluble, okay, and the GC is then for guanylate and cyclase. Okay, right, so this is very similar to an adenylate cyclase enzyme or an adenylyl cyclase enzyme, except that instead of doing it producing cyclic AMP, they produce cyclic GMP. So instead of adenosine, it's guanosine, hence why this is guanylyl or guanylate rather than adenylate or adenylyl. And you could call this soluble guanylyl cyclase if you liked. It's more commonly referred to as the soluble guanylate cyclase, but guanylyl and guanylate mean exactly the same thing. Okay, and unlike the adenylyl cyclase enzymes, which are in the cell membrane, and they have those incredibly impressive structures with 12 membrane-spanning alpha helices, the soluble guanylate cyclase is just in the cytoplasm of the cell, hence why it's called the soluble guanylate cyclase. So it's just floating around in the cytoplasm of the vascular smooth muscle cells, and when nitric oxide comes along, it's going to activate this enzyme. And as I say, this enzyme is now going to take guanosine triphosphate molecules, GTP molecules, and break them down into pyrophosphate, which is two phosphate groups linked together and is written PPI, and also of course cyclic GMP, which is the important product here, abbreviated down to CGMP. So, the nitric oxide causes a rise then in the level of cyclic GMP within the vascular smooth muscle cell cytoplasm. Cyclic GMP then activates an enzyme known as protein kinase G, which is very similar to protein kinase A, except that it's activated by cyclic GMP, and protein kinase A is obviously, of course, activated by cyclic AMP. So this now is PKG for short, and this stands for protein kinase G. You might also hear it referred to as the cyclic GMP dependent protein kinase. That's just its full name. Everyone calls it protein kinase G. Okay, so in pink here we have protein kinase G, and this is a serine-threonine kinase enzyme. It adds phosphate groups onto proteins by adding them onto their serine and threonine residues at phosphorylation sites, and therefore modifies the function of proteins. And protein kinase G is specifically now going to phosphorylate and inhibit the final enzyme that I'm going to talk about in this cascade, which is the myosin light chain kinase, the MLCK enzyme. Now this is a really important enzyme inside smooth muscle cells. Okay, so I'll firstly write down its name fully, and then I'll explain how important it is. We're not going to take the pathway any further, but I will explain to you just how important myosin light chain kinase is. Okay, so myosin light chain kinase, it is effectively the control switch for whether a vascular smooth muscle cell is going to contract or whether it's going to relax. Myosin light chain kinase, it turns on the contractile machinery within the vascular smooth muscle cell. So the vascular smooth muscle cell, its cytoplasm is full up with contractile machinery, okay, contractile fibers that are capable of contracting the vascular smooth muscle cell down. To turn that contractile machinery on, to make the vascular smooth muscle cell contract, you need to turn on myosin light chain kinase. It is the switch for contraction. It goes around and phosphorylates the contractile machinery, different proteins of the contractile machinery, in ways that we're not going to go into the details of because it will take me half an hour to explain it all, okay? But the important thing to understand is it's the switch. It goes around and turns on all the contractile machinery and causes the vascular smooth muscle cell to contract. If it is turned off, therefore, the vascular smooth muscle cell is also, as far as contraction is concerned, turned off. So protein kinase G is going to turn off lots of myosin light chain kinase enzymes, and therefore it's going to cause relaxation. So the contractile or relaxed state of the vascular smooth muscle cell is determined by how many myosin light chain kinase enzymes are on and how many are off. And protein kinase G becoming activated is going to turn a whole bunch of myosin light chain kinase enzymes off, and therefore we're going to get relaxation of the vascular smooth muscle cell. So as I say, we're not going to go any further with that and discuss exactly how this controls the contractile state. We're just going to state that it does. Okay, right, so to summarise then, endothelial cells which are exposed to too high shear stress, they know that there is something wrong with the diameter of the lumen of the blood vessel. They're experiencing too high shear stress, so the lumen of the blood vessel can't be quite right. So what they do is they release this signalling molecule, this call for some sort of cooperation on the part of the vascular smooth muscle cells. They send this signalling molecule, nitric oxide, to their surrounding vascular smooth muscle cells, saying, we need you to relax down because the lumen needs to become larger. Okay, the blood is having to move too fast through the lumen, therefore we need the lumen size to increase. Nitric oxide works in the vascular smooth muscle cells by activating the soluble guanylate cyclase, which produces cyclic GMP, which then activates protein kinase G, which turns off a bunch of myosin light chain kinase enzymes, and that is the switch for the contraction relaxation state. So if we turn it off, we're going to get less contraction, more relaxation. So the vascular smooth muscle cells will all increase in length, and the circumference of the vascular smooth muscle cell rings will all increase, and therefore the diameter of the vascular smooth muscle cell rings will all increase, and therefore the diameter of the lumen of the blood vessel will increase. So, coming back to our occluded coronary vessel then, this pathway will be activated continuously, and therefore what you will be doing continuously is relaxing down this portion of the blood vessel, okay, relaxing its vascular smooth muscle cells to try and increase the lumen back up. So this will permanently dilate to try and increase the blood vessel lumen, and therefore try and maximize that diameter. So the important point here is that it can't dilate any further. When you've got this chronic atherosclerotic plaque in your coronary artery, whichever one it is, then the surrounding vascular smooth muscle cells will be as dilated or as relaxed as they possibly can be, and the blood vessel will be in this permanently dilated state to try and maximize the size of the lumen, and therefore maximize the size of the hole that the blood is having to flow through. Okay, the important thing for you to understand is that there is no extra wiggle room. It can't dilate any further is the problem. Whereas the healthy coronary arteries, they are not in this permanently vasodilated state, and they can vasodilate more. Now, but when you are at rest, this works absolutely fine. Your coronary artery may have this atherosclerotic plaque in it, but because of the fact that it's continuously in this permanently dilated state, you are getting an okay amount of blood through it, and this is still supplying, you know, the heart muscle perfectly well. But now let's think about what's going to happen when you start exercising. So firstly, it helps to think about what should happen when you exercise in a healthy person. Okay, so let's say we begin exercising. Let's draw a little flow diagram for this. Okay, so I'm a healthy person, I do not have atherosclerotic plaques in my coronary vessels. What happens when I start exercising? Well, of course, when you start exercising, you're using your skeletal muscle cells a lot more than normal, and they obviously need blood going to them in order for them to maintain their activity levels. Okay, so they're going to need oxygen and glucose arriving. Specifically oxygen, because of course they have huge glycogen reserves, so they're okay on glucose, but they need oxygen. So you're going to need to increase cardiac output. So in exercise, cardiac output has to go up. The amount of blood that you're pumping around the body in a minute needs to go up. To get cardiac output to go up, that means the heart's going to have to work harder. Now, how does the heart work harder? Well, it increases the rate at which it beats, and it also increases the force of contraction. In order to achieve this, what actually happens is the sympathetic nervous system increases its stimulation on the heart. So the SNS increases its stimulation on the heart. And I realise this is a bit of an odd flow diagram, because really the cardiac output going up should be the last result. So in fact, let me get rid of this. This is our aim, okay? So I won't get rid of it, I'll just replace it. The aim is to raise cardiac output. How are we actually going to achieve that? The sympathetic nervous system is going to increase its stimulation on the heart. So you're going to release noradrenaline in a greater amount now onto the myocardiocytes. And of course, noradrenaline will act on beta-1 receptors on the surface of the cardiomyocytes, and it will act all over the heart. And when beta-1 receptors are stimulated on the sinoatrial node cardiomyocytes, what this will lead to is increased rapidity of the firing of those action potentials that the sinoatrial node cells generate spontaneously, so you're going to get increased heart rate. And when beta-1 receptors are activated all over the heart on the actual contractile cardiomyocytes, of course the effect that it has is it causes those cardiomyocytes to contract harder, to generate more force. So whenever they get stimulated to undergo contraction, they're going to contract harder, they're going to generate more force each time they actually do undergo the contraction. Okay, so that's known as inotropy going up. So inotropy refers to how hard the heart is actually beating, how much force each cardiomyocyte generates when it actually receives the electrical signal to contract and does contract. So the heart rate goes up and all the cardiomyocytes start working harder every time they do contract. So they're having to contract more times each minute, and they're having to work harder every time they do contract. And of course this is fantastic for the entire body, because it's going to mean that they're going to eject a greater volume of blood every time they actually do contract. So this will increase stroke volume, so the actual amount of blood that is ejected into the aorta every time the heart does beat is going to increase. And of course this means that the heart is beating more times every minute, and both of these together, both the fact that we're beating more times a minute and the fact that every beat is ejecting more blood, is going to then lead to cardiac output going up. And this is fantastic, because it means that the skeletal muscle cells are now going to get the oxygen that they need. Okay, now this isn't so good of course for the heart, because the heart muscle cells are now having to work much harder. They're having to contract more frequently because heart rates increase, and they're having to work, when they do contract, they're having to contract harder than before. So they're going to require a greater supply of oxygen themselves. Okay, so we're going to have the blood flow to the cardiomyocytes increased. Now, how do we actually increase the blood flow to the cardiomyocytes? Well, we dilate all of the coronary blood vessels. So in fact, sympathetic stimulation onto the vascular smooth muscle cells of the coronary arteries, it acts on beta-2 receptors on the surface of the vascular smooth muscle cells. So I'll just draw this here. So in the vascular smooth muscle cells of the coronary arteries, you have beta-2 receptors, And when the noradrenaline acts on beta-2 adrenaline receptors, it activates the alpha-S G proteins, okay, or the GS, heterotrimeric G protein, which then activates adenylyl cyclase, which then activates protein kinase A. And protein kinase A then does the same thing as protein kinase G. It inactivates myosin light chain kinase, and therefore leads to the relaxation of the vascular smooth muscle cells. So this is going to act on beta-2 adrenoreceptors on the surface of the vascular smooth muscle cells, and this will cause vasodilatation in the coronary arteries. Now that's rare, because usually when you stimulate vascular smooth muscle cells with noradrenaline or adrenaline, it causes contraction of the vascular smooth muscle cells, and that's because there's usually a different receptor on vascular smooth muscle cells. Usually you have the alpha-1 adrenoreceptor, which causes contraction when noradrenaline acts on it in the vascular smooth muscle cells. In the vascular smooth muscle cells of the coronary arteries, you have the beta-2 adrenoreceptor instead, and this causes a different signaling pathway to be activated inside the cells, and it leads to relaxation, and therefore you get vasodilatation of the coronary arteries, and therefore increased blood flow to the heart, and this is why the heart can actually sustain this higher workload. Right, now in our person with an atherosclerotic plaque in their coronary artery, of course we know that the coronary artery at that point of the atherosclerotic plaque is dilated as much as it possibly can be because of the presence of the atherosclerotic plaque. So it can't dilate any further, so you can't increase blood flow through that artery any further. So therefore you don't get increased blood flow to the portion of myocardium that artery supplies, even when you start exercising, and that's why you're going to get the problems when you start exercising. So when you're resting, it's okay because your coronary artery is dilated as much as it can be at that point of the atherosclerotic plaque because of this pathway that we've discussed here. So blood flow through the coronary artery is salvaged basically because of the fact that you're dilated to the maximum amount that you can be. So even though you've got the atherosclerotic plaque, it doesn't matter. However, when you start exercising, then you need dilation even further, but that portion of the coronary artery with the atherosclerotic plaque in it's already dilated as much as it possibly can be. It can't dilate any further, so you don't get increased blood flow to the area that blood vessel supplies, even though you've started to exercise, and that's what's going to cause the problem. That's why you're going to get crushing chest pain. So now let's actually discuss, where does the crushing chest pain come from? So if you have this coronary artery disease, then you are now going to get ischemia. So ischemia does not mean full cessation of blood flow to an area. Okay, this is a common misconception that ischemia means no blood getting to a certain area. Ischemia just means that there is not enough blood getting to an area to meet the demands of that area. So indeed, this is what's going to happen. So if I go back to my picture here, if we've, say, got an atherosclerotic plaque in this portion of the left anterior descending artery, then, as I say, when you're at rest, it's fine because the coronary artery will have dilated around that point as much as possible, so you'll have normal blood flow, or as normal as possible. But when you start exercising, it's going to be a problem because now you're still going to just get the same amount of blood coming here, even though you need more blood now getting here because the heart's going to work so much harder. Okay, so now this portion of tissue is not going to receive the amount of blood it needs to be receiving for how much it's demanding. That mismatch in the amount of blood that's actually getting to an area and its demand for blood, that's called ischemia. So when the demand for blood perfusion is greater than the amount that's actually getting there, that's what's known as ischemia. It does not necessarily mean that you have to have no blood flow to an area. So this portion down here, supplied by the left anterior descending artery, this is going to become ischemic in this case when we start exercising. Okay, so the ischemia then of this portion of myocardium is then what's going to lead to the crushing chest pain, which is known as the angina pectoris. Okay, so why does it actually lead to the crushing chest pain, and also why do we get referral of the pain up to the left shoulder? That's the question that I want to now ask. Well, why does it lead to the pain then is the first question. Well, there is still a little bit of debate about exactly why it causes pain, but I think the general consensus is that the answer is that when a tissue becomes ischemic, it has to turn on anaerobic respiration far more. And anaerobic respiration produces lactic acid. Lactic acid is an acid and donates protons away into solution, and therefore you get a drop in pH. You get what's known as lactic acidosis, and that is what activates the pain neurons. So let me just talk about that in a little bit more detail. So let me remind you of the basics of respiration. So I'm not going to go through the full respiration reaction because it is absolutely enormous, but I'm just going to summarize what normally happens in aerobic respiration and then what normally happens in anaerobic respiration. So let's start with aerobic respiration. And remember, we are talking about the process inside a single cell, so we're talking about the cellular process of respiration. Okay, so aerobic respiration. So remember, in aerobic respiration, there are four main stages. Glycolysis is the first stage, and in glycolysis what happens, and I'll just make sure this is straight, excellent, yes it is. In glycolysis what happens is you take a glucose molecule, and through a huge number of steps, the glucose molecule is converted into two pyruvic acid molecules. Okay, so we get two pyruvic acids. Okay, so that is the process of glycolysis. That arrow summarizes what glycolysis does. Then what happens is these pyruvate or pyruvic acid molecules, these are going to go through a small reaction known as the link reaction, which then produces two acetyl coenzyme A molecules. Okay, so we're then going to end up with two acetyl coenzyme A molecules, and this also transports the molecules across into the mitochondrial matrix, because remember glycolysis, and I'll just add a bit of colour on here, so we'll have glycolysis coloured in blue here. Glycolysis occurs in the cytoplasm, whereas the Krebs cycle, the next stage, is going to occur in the matrix of the mitochondria. So this intermediate reaction here, known as the link reaction, this transports the molecules into the matrix of the mitochondria. So this is the link reaction, and I'll just colour this in orange like so. Okay, then what's going to happen is the two acetyl coenzyme A molecules, they are going to go through the Krebs cycle here, and of course the Krebs cycle will completely destroy them. It will turn the carbons of the acetyl coenzyme A molecules into carbon dioxide. The link reaction also actually produces a molecule of carbon dioxide, because those of you who know the chemical structures of these molecules will know that pyruvic acid has 3 carbons, acetyl coenzyme A only has 2 carbons in the actual acetyl. I don't know how many carbons are in the coenzyme A, but that's just an accessory thing that's been added on. Okay, so the 2 remaining carbons here, however, are going to be going off in carbon dioxide, and the main product that we end up with here is these reduced electron carriers. So we end up with reduced NADH, and we end up with also reduced FADH. Okay, and we end up with lots of these. And these reduced electron carriers, and we, by the way, also end up with one of these from glycolysis. So glycolysis also produces 2 molecules of reduced NAD as well. And also, actually, I'll add this on as well, it also produces 2 molecules of ATP on its own. And that's actually why it can be used in anaerobic respiration. So we'll come back to this. So glycolysis, overall, what does it do? It turns glucose into 2 pyruvic acid molecules, and these pyruvic acid molecules can go through a lot of other steps, providing we have lots of oxygen for aerobic respiration, but on its own it produces 2 ATP molecules and 2 of these reduced NAD molecules. The reduced NAD here, the electron carrier, and the reduced electron carriers that we get from the Krebs cycle, which I'll colour in pink over here, these can now all go on to the electron transport chain. So I'll draw this here, so we'll have just ECT, like so, and of course the electron transport chain then chucks out absolutely loads of ATP. Okay, so this results in a huge amount of ATP here. Right, so that's my brief summary then of aerobic respiration. So glucose, we start off with that, then it goes through the process of glycolysis, which occurs in the cytoplasm, where we convert it into 2 pyruvic acid molecules and 2 ATP molecules and 2 NAD molecules that are reduced. The 2-ATP, of course, they're ready for use, fantastic. The reduced NAD, we can get lots of energy from that using the electron transport chain. We can take the pyruvic acid molecules further through the link reaction, then through the Krebs cycle, and this will result in loads of reduced electron carriers, both at the NAD variety and the FAD variety. All of these reduced electron carriers then go through the electron transport chain, and that results in a huge amount of ATP to be generated. Now, the important thing is the electron transport chain requires oxygen. Okay, so you can only do this if you have a lot of oxygen. If you don't have a lot of oxygen, then all of this backs up, and it would halt the entire process, starting with glycolysis. However, there is an escape route for glycolysis to go down. So when pyruvic acid molecules start backing up, they start building up because the whole process is lagging because of the lack of oxygen, what happens is they can be instead converted into lactic acid molecules. So both of these pyruvic acid molecules can be converted into lactic acid molecules. And the idea here is that we're going to just use the ATP molecules generated by the glycolysis process. So in anaerobic respiration, what you do is you do glycolysis exactly the same. So glycolysis forms part of the anaerobic respiration system. Okay, so you turn the glucose molecules into the 2 pyruvic acid molecules. You produce 2 ATP, fantastic. That's the energy that's produced by anaerobic respiration. But then we need to do something with these silly pyruvic acid molecules and the reduced electron carrier here, because if we want to continue doing more and more glycolysis to get the ATP that we're getting from it, okay, which is what the only source of ATP in anaerobic respiration is, we need to regenerate the oxidized form of the electron carrier. We need to get rid of the electrons that this carries, and also, of course, the proton that it carries. So, what we do is we give it back to the pyruvic acid molecules and convert them into lactic acid molecules. So I'll just get another piece of paper and I'll actually draw out the chemical pathway here. So, let me just show you the structure of a pyruvic acid molecule and then a lactic acid molecule, and I'll explain to you how we can reduce the pyruvic acid molecule into a lactic acid molecule. So we'll start off with pyruvic acid, which I'll draw here. Okay, so as I say, pyruvic acid is a 3-carbon molecule, and we're going to have a carboxylic acid group, which is obviously why this is an acid. Then we've got the other 2 carbons here, and the structure is very simple. We're going to have a carbonyl group in the middle, like so, and then a methyl group at the end here. So there is the structure of pyruvic acid. Okay, so we end up with 2 of these at the end of glycolysis, and we want to somehow regenerate the oxidized form of the electron carrier, NAD, nicotinamide adenine dinucleotide. Okay, so let me just remind you of how NAD works. So NAD is a molecule that can accept electrons, and normally it has a positive charge, so we refer to it as an electron carrier. Now, how can it accept electrons? Well, what it can actually accept is what's known as a hydride anion. Okay, a negatively charged hydrogen atom. Now, you'll have been used to seeing hydrogen ions, but the hydrogen ion that you'll be used to seeing is not the hydride anion, it's the hydrogen cation. Now remember, hydrogen's a very simple atom. It just consists of a proton with an electron orbiting it. In the hydrogen cation, the hydrogen atom loses its outer shell electron and just becomes a proton. In the hydride anion, what happens is it instead accepts an electron, gets 2 electrons in its outer shell, is very happy at having paired its electron up with something, and now has a negative charge. So a hydride anion consists of a proton with 2 orbiting electrons. Now, nicotinamide adenine dinucleotide in the oxidized form here can now accept one of these hydride anions, like so, and become NADH, or the reduced nicotinamide adenine dinucleotide. And this will be carrying, effectively, 1 proton and 2 electrons. However, often we think of this as working with an additional hydrogen cation that is in free solution. So hydrogen cations, which are just the protons, they're very easy to come by. Okay, hydride anions are not easy to come by. Hydrogen cations are very easy to come by. They're just in free solution. So often when reduced NAD molecules are actually going to give up their hydride anion to a molecule, they work alongside a proton from free solution. So this is why people often put NADH plus a proton in this way. Okay, the proton comes from free solution, the hydride anion comes from the electron carrier, and together really they're providing two hydrogen atoms, because this hydride anion that the electron carrier provides can give its additional electron to the hydrogen cation, and therefore you're effectively providing two hydrogen atoms. So really, for the basis of actually using electron carriers to drive reactions, you can think of them both, and this is both nicotinamide adenine dinucleotide and also nicotinamide adenine dinucleotide phosphate. With FAD, flavin adenine dinucleotide, it's slightly simpler because that really does just carry two hydrogen atoms. But with these two, NAD and also NADP, it's more complicated. They just carry hydride anions, but in effect you can think of them as carrying two hydrogen atoms because they can just snatch a proton from free solution, and then effectively they've donated to a chemical system two hydrogen atoms. I hope that makes sense. So in this reaction now, what's going to happen is the reduced NAD with a proton is going to provide two hydrogen atoms into this molecule, and that's going to convert it into lactic acid. Okay, so this reaction is carried out by lactate dehydrogenase, and the reason it's called that is it's named for catalyzing the reverse reaction of this arrow. So the reaction is actually reversible. So I'll explain the name in just a moment. So let me now actually show you the structure then of lactic acid. So what we're going to do, this isn't an electronic flow diagram, but this will help you to understand it. We're effectively going to break one of the two bonds in that double bond between the carbon and the oxygen in pyruvic acid here. And you can imagine sending one electron back to the carbon, one back to the oxygen. So remember, in a covalent bond there are two electrons in that, one from each of the two members of the covalent bond. So imagine sending the electron from the carbon back to the carbon, the electron from the oxygen back to the oxygen. Then what we're going to do is we're going to take our two hydrogen atoms, which have been provided by this electron carrier in the way that I have explained to you now in detail, i.e. the hydride anion is the bit that's actually carried by the electron carrier here, which is NAD, and the proton just comes from free solution. And then we're going to put a hydrogen atom onto the oxygen and a hydrogen atom onto the carbon, and what we'll end up with then is, here is the carboxylic acid group, like so. Here's this carbon, we'll have an alcohol group now here, a hydrogen here, and then we'll have our methyl group here. And this then is the molecule of lactic acid. So the reason the enzyme that catalyzes this is called lactate dehydrogenase is because it's named for catalyzing the reverse reaction here, where lactic acid goes to pyruvic acid, and of course there you're removing two hydrogen atoms, so it's a dehydrogenase reaction. Okay, right, but of course in our case, in the case of anaerobic respiration, we are interested in it going in the forwards direction here. So in anaerobic respiration then, what's going to happen, we'll need this here, going back to this picture, is that you're going to participate in glycolysis. The glucose is going to go to the two pyruvic acids. That generates us two ATPs. That's where the energy comes from in anaerobic respiration. We then need to get rid of both of these reduced electron carriers. We need to return them back to the oxidized electron carrier so that we can continue on with glycolysis. So we give the hydride anions that these two carry to the two pyruvic acid molecules and convert them into two lactic acid molecules. And now the lactic acid just builds up and up and up. What's the problem with that? Well, of course, lactic acid can donate its protons away into solution. So coming back to the picture of lactic acid here, this carboxylic acid group of the lactic acid molecule, this is capable of donating that proton off the oxygen away into free solution, and therefore is capable of lowering the pH in the area. So the extracellular fluid effectively in the ischemic portion of the myocardium is going to become more and more acidic. The pH is going to go down and down as the proton concentration goes up. Remember, pH has that weird relation with proton concentration. So as proton concentration goes up, pH goes down, it becomes more acidic. Okay, and becoming more acidic is not good. Pain neurons are very good at picking up problems with the pH of the extracellular fluid, and they're activated to fire action potentials by the extracellular fluid being too acidic. So let me write this down. So when pH goes too low, what that is going to cause is it's going to activate the endings of primary nociceptive neurons, as they're called. Okay, so it's going to activate primary, which just means they're the first neuron in the pain pathway. Nociceptive, which is a fancy word for pain. Nociception is the perception of pain. So primary nociceptive, and then it's afferent because it's going into the spinal cord. So primary nociceptive afferents are the fancy word for these first order pain neurons that are going to respond to the pH going too low. Okay, and that's then going to convey the information up into the central nervous system, and that's going to create the conscious sensation of horrendous crushing chest pain in the heart upon exercise. So the final thing that I want to explain then is why do you get the referred pain up to the left shoulder? Well, this has to do with the convergence of primary nociceptive afferents onto secondary spinothalamic neurons. So, the primary nociceptive afferents, they are activated by the pH being too low in the extracellular fluid. They're going to conduct action potentials and carry the signal up into the spinal cord, but then they pass the signal on to another neuron. And the thing that causes referred pain is that multiple different primary nociceptive afferents can actually synapse onto the same second order neuron. So let me just draw a picture of this. So what I'm going to do is I'm going to draw a picture of the spinal cord here. Okay, so I'm going to have a cross section through the spinal cord. So here we go, like so, and this picture isn't looking brilliant, but never mind. Okay, so here is the anterior median fissure, and then we'll have the posterior median sulcus. Then I'll mark on the grey matter of the spinal cord here, which has this characteristic shape like so. The ventral horn, another ventral horn, the right ventral horn, and then, oh dear, it's gone awful at the end, hasn't it? Never mind. Here, hopefully you can appreciate this is the spinal cord, and let me just remind you of a few of the key structures here. So this crease inwards, this is known as the posterior median sulcus. So this is just to orient you. This is the posterior portion of the spinal cord. So I've taken a transverse cross section here. This is the posterior bit. So this is the posterior median sulcus. Okay, of course these are the two dorsal columns of white matter. This fissure in the front of the spinal cord, this is known as the anterior median fissure of the spinal cord. So this is the front of your body, if you like, or the, well, not the front of your body, but the front of your spinal cord. This is facing the front of your body. Okay, and I'll just add a little bit of colour on here. So we'll have this coloured in green, and we'll have this coloured in blue here. Then what we have, of course, marked here is the grey matter, like so. So we have the two ventral horns, the right ventral horn, so this is the right side, this is the left side, so this is the left ventral horn. Then we have the posterior horns, or the dorsal horns, so this is the left dorsal horn, and this is the right dorsal horn. White matter columns, we have the lateral column here, this one, of course, the left lateral column, this one, the right lateral column. We have the anterior white matter columns here, left anterior column, right anterior column, and then the anterolateral columns, these little spaces here. Okay, left anterolateral column, right anterolateral column. Okay, so that's just to orient you. Now, of course, let's draw on the spinal roots. So, of course, we have two roots on each side of the spinal cord coming off all the way up and down the spinal cord. So let's say we have been lucky enough to take this cross section at a portion where we do indeed go across the roots coming out of the spinal cord. So I'll draw roots on, and I'll just do it on the right side here rather than on both sides. So here is the ventral root coming out like so. Then we'll have the dorsal root here, and I'll have the dorsal root ganglion as this little bulge here, like so. And then the ventral and dorsal roots will then combine together to form a mixed spinal nerve, like so. Okay, right. Now, you'll remember from basic anatomy that the ventral root here, this is going to contain outgoing fibres, efferent fibres. So, for instance, alpha motor neurons that are going to synapse on skeletal muscle cells all over the body. These will come out in the ventral root here and then join the mixed spinal nerve. Whereas afferent fibres that are coming into the spinal cord, such as sensory fibres, indeed pain fibres, they're going to have their processes going up the dorsal root here. So indeed, my primary nociceptive afferent, which is of interest to me, its going to come up like this. So here is its axon, and it's going to have its cell body in the dorsal root ganglion here. So this is meant to represent the dorsal root ganglion, the DRG. So here is its cell body, and then its central process is going to go in. So the primary nociceptive afferents look like this. They are known as pseudo-unipolar cells. So they have their cell body like so, then they have a single process coming off, but this splits into two like so. So it will have one that goes peripherally, and this is where you had the receptors for acid, and they were activated, and this caused an action potential to fire up here. So this is the peripheral process. Okay. And then we've also got the central process, which is now going to deliver the action potential into the central nervous system, and is going to have a synapse with a next neuron along. And here, of course, is the cell body. So this sort of a neuron where you've just got a single process coming off, but really you've got two processes, because this single process splits into two processes with very different functions. This is known as pseudo-unipolar. So these primary nociceptive afferents are described as pseudo-unipolar neurons. Okay, right. So, the pH then, going down, stimulated the peripheral process of this primary nociceptive afferent far, far away in the heart somewhere. Okay, now the action potential is coming up, it goes down the central process here, and then it doesn't deliver it all the way up to the brain. Instead, what happens is the primary nociceptive afferent is going to have a synapse with a secondary nociceptive neuron here, which is known as a secondary spinothalamic neuron. So a secondary spino, because it's going to go from the spine up to the thalamus in the brain, so spinothalamic neuron. Okay? And this secondary spinothalamic neuron is then going to fire an action potential if it receives enough stimulation, and it will fire this action potential along its axon like so, which will cross over onto the contralateral side and will ascend up in the anterolateral column on the contralateral side, and will go up to the thalamus on the contralateral side, and then will be delivered to the rest of the brain. Okay, right, so we want to try and explain then the phenomenon of referred pain to the left shoulder. Well, the phenomenon of referred pain to the left shoulder occurs because of this synapse here. So it is not the case that there is a one-to-one correspondence between primary nociceptive afferents and secondary spinothalamic neurons. Okay, so each of these primary nociceptive afferents, and I'm going to colour them in separate colours. So there's the secondary nociceptive afferent in blue, and now I'll colour in the primary nociceptive afferent here in orange. Each of these primary nociceptive afferents bringing in information about pain all over the body and that are coming into the spinal cord, they do not all have their own secondary spinothalamic neuron to synapse with. Loads of them have to share. So loads of them synapse onto the exact same secondary spinothalamic neuron. So if this secondary spinothalamic neuron fires an action potential, you do not know which of the primary nociceptive afferents actually stimulated it to fire an action potential. Now the reason then that you get referred pain in the case of angina to the left shoulder is because primary nociceptive afferents from the heart are going to synapse onto secondary spinothalamic neurons that are also used by primary nociceptive afferents from the left shoulder. Okay, so you're going to have primary nociceptive afferents coming from the heart and also primary nociceptive afferents coming from the left shoulder, and they will now both be sharing the same secondary spinothalamic neuron, so they'll be stimulating the same secondary spinothalamic neuron. So when this secondary spinothalamic neuron fires and stimulates centers up in the brain, those centers up in the brain do not realize that that is being stimulated by the heart. They think it must be stimulated by the primary nociceptive afferents that have come from the left shoulder rather than the heart, because it's not a normal thing to have pain sensations coming from the heart. It's far more normal to have pain sensations coming from the left shoulder because some other being in this world has attacked you and hurt your left shoulder. That's far more normal. So when the brain gets signals coming from the secondary spinothalamic neurons, it thinks that they must have been stimulated by the primary nociceptive afferents from the left shoulder rather than from the heart. And that's why, even though the pain signal is coming from the heart, the brain perceives it as coming from the left shoulder, and why the conscious sensation that you then get is the same as having pain in the left shoulder. Now, of course, not all of the primary nociceptive afferents that are coming from the heart will share secondary spinothalamic neurons with primary nociceptive afferents from the left shoulder, which is why you still do get some, indeed a lot of pain that is evidently coming from where your heart is, but you're also going to get some pain coming from the left shoulder, or at least perceived to be in the left shoulder, and that's from these primary nociceptive afferents from the heart, which synapse onto the same second order spinothalamic neurons as primary nociceptive afferents from the left shoulder. Okay, so I hope you understand why you also get this referred pain then from the left shoulder when angina pectoris strikes. Okay, so usually this will relapse when you stop exercising, so normally the huge crushing chest pain is enough to make people stop exercising, and when they stop exercising, of course, their heart rate will go back to normal, their contractility of the cardiomyocytes will go back to normal, the sympathetic stimulation on the cardiomyocytes indeed will go back to normal, causing those two things, and therefore the demand for oxygen of the cardiomyocytes will go back down, and therefore ischemia will no longer be a problem because the blood flow is adequate for the cardiomyocytes when they're in the normal resting state and not having to work at this ridiculous level, and therefore the lactic acidosis will be cleared up once there's no longer generation of more lactic acid, and the symptoms will reside. What we'll talk about then in the next video is acute coronary syndromes, which are acute escalations that can happen when atherosclerotic plaques in coronary arteries rupture. We will come back to the treatment of angina pectoris, because when someone is suffering from stable angina, there is a huge risk that the atherosclerotic plaques in their coronary arteries that are causing the stable angina are actually going to rupture, and then they will have an acute coronary syndrome. So we give people with angina pectoris a lot of treatment to try and prevent them actually having an acute coronary syndrome, and of course that's the major acute coronary syndrome is heart attack, and that's what we're trying to prevent. But we'll come back to the treatment, because as I said at the start, it's difficult to explain the mechanism of actions of drugs that are designed to stop heart attack without first looking at what happens in a heart attack. Okay, so we'll do that in the next video.